Hi guys, so basically today we are going to be taking a look at the Camel-ai's optimized workforce learning framework, and then we're going to be implementing a sort of like a little project where essentially we're going to be navigating to a website which hosts like cellular imaging data, and then we're going to be filtering out those data sets based off of our specific requests. So for example, if you would like to take a look at a data set of a specific protein, we can get those data sets, and then we can gather some information and print it and export it to the user in the form of a response from the AI agent. So basically, with the help of OWL, the idea that was floating around in my head is that I wanted to implement with the help of the Camel-ai's OWL framework an AI agent that was going to be searching and analyzing protein tomograms from the CZI, the Chan Zuckerberg Imaging Institute Cryo ET data portal, using obviously the Camel-ai's OWL framework. So it's a bit repetitive, but yes, that's how prominent OWL framework is. So basically, what exactly do we have? We have a bunch of directions on how to run this repository, and then there is a repository that hosts the project source files. So yes, in order to even be able to work with the OWL framework, you need to create an environment for yourself with the help of a package manager and a channel. So in this case, I'm using the Conda Forge channel and a package manager Mamba. So, and then for the models, the AI models, I'm going to be using the Gemini, Google's Gemini 2.5 Pro experimental, in order to navigate to the page and gather insights about the certain data sets. And then we are going to be installing certain requirements, certain packages, and the browser window in this case that OWL spawns are from a package called Playwright. So yes, Playwright, make sure you have Playwright installed. So right now I'm just going to be activating my environment, which is Camel environment, and for testing purposes we are going to be isolating other environments. For example, if you're testing out with OWL separately, you can have another environment initiated for that. So we have a version Python 3.12.9. Camel-ai's OWL officially supports Python 3.10, 3.11, 3.12. So make sure you do have a version between them. And right now we are installing just dependencies with the help of requirements.txt. This might take a while, so we will skip 30 seconds ahead. And then quickly after the packages were installed, we are now testing out whether the browser window is going to be able to spawn or not with the help of Playwright, to make sure that Playwright is successfully installed and is not having any issues. So we are going to be navigating to the browser, and then we are going to be navigating to the page where the datasets are hosted on the Cryo ET data portal, and then that would suffice and give us enough of an indication that we can now navigate to the page and search for certain datasets ourselves with the help of this AI agent. So it's taking a while. It's right now giving us a blank screen, but eventually this Chromium instance is going to launch the page where we have our datasets. And as you can see, we are on the Cryo ET data portal, and then we have a bunch of datasets that are by default displayed. And in the background there are certain tests further which have not completed due to certain rate limits on my models, because I'm using Grok API for them. You can also use the Grok API to run certain models, open source models as well. Right now we only want to deal with the Gemini API and the Gemini suite of models, because they by far are going to be the best in terms of basically having you up and running with the OWL framework pretty quickly. So that's why yes, I decided to go with the Gemini models. So now exactly we know that the browser is working. Now we can see and search for a tomogram of Salmonella. Salmonella is an organism that is responsible for various fevers. We have a virus that is Salmonella typhi, which is responsible for typhoid fever. So we're going to be running all of the data. We are going to be searching for all of the data sets that have the name Salmonella in them. So we ran our file python run.py, and then we gave it our query, which is Salmonella. So we have a communicative role-playing AI agents in the background. So we have our in the communicative function, we have our in the agent construction function, we have our task prompt, and then after that, we in between the OWLs we have a AI user agent, and then assistant agent, and then a bunch of agents in the OWL framework as well that are going to be launching this browser window and navigating to the page, searching for the relevant fields where you can put the data, such as searching for the input fields, and then we can provide a text there to filter out the results. So yes, now we are finally on the page, and then this is completely autonomous. I'm not doing anything, I'm not even touching my mouse. So all is being done by the OWL framework, and basically in the background AI agent is giving specific instructions to what to do next to the OWL framework exactly. So as you can see, we have now filtered out the results on the dataset page and then searched for all of those datasets that have the term salmonella in them. And now what we are going to be doing, we are going to be taking a few of these dataset cards, compiling them, and then essentially gathering some insightful information from these datasets. So we can get metadata, we can get their authors, we can get their runs, and then we can get how annotated these datasets are, or exactly how much highlighting these datasets do have, where, in which parts of these tomograms for highlighting do we have. So for example, if an organism is specifically present inside a tomogram, its borders, or at least its edges, only might be annotated, only might be highlighted for specific use cases. So yes, there's that. We can also modify and filter out the results based on the annotations as well. So now we got this huge response, which is just a massive blob of text, and then we only have included the first 5 results from this dataset query. So we are going to be just taking a look at the information from a single dataset card, and then by inputting this data in a text editor such as NeoVim, we are going to be gathering a specific line for this dataset card, and then we are going to be gathering what exact data did our AI agent pull for each dataset card containing the term salmonella in them. So let's take a look. We have a massive blob of text. We are going to be navigating to this dataset card titled Salmonella Flil mini cells, and then we just going to be quickly taking a look in a while. So let's quickly get to the to our title for a particular dataset cards. There are a bunch of dataset cards that have been pulled by our AI agent, but we only want to see exactly how much information did we pull and how much of it can be exported by further analysis. So we're going to be taking out, we are going to be taking a look at this Salmonella Flil knockout mini cells. So we have this title, description, which was not entirely visible on the card, so our AI agent put that it's not visible, metadata, dataset, ID, authors, and then organism inside of this tomogram. We basically copy this line and then we format it down below so that it's much more readable. So we can go down below and then we can paste up a bit to get the results. So we get our AI agent collected information about tomograms autonomously containing the term salmonella in them, and the information it came up with for each of the tomograms are title, description, metadata, authors, and the organism inside of the tomogram, and then the runs, which is also a field on the dataset page, annotations, and the URL associated with that dataset card. And in the fields, it could not automatically detect the information behind it. It inputted it with a text that the exact thing could not be found, but it was indeed present, but was not visible for some reason. So yes, this was the project. Hopefully you guys had a lot to learn from OWL's library, and then if you do want to take a look on this project further, you can navigate to this link, github.com/developcoder/cryo tomogram search agent, where we have hosted the source files. So yes, this was an interesting project to be a part of. Thank you.